Hello friends, welcome back to my channel. In previous lecture we discussed about infection, and in this lecture we discuss about bacterial toxins, how bacteria produce toxins and cause disease in human being or host. So let us continue this lecture. Toxigenicity. What is toxigenicity? Toxins are small molecules and peptide or proteins molecules which produced by microorganisms, and these toxins cause harm to host and causes disease. Frequently, these toxins are the main pathogenic factor of microorganism. Toxins effect may include fever, cardiovascular problem, diarrhea, shock, destruction of RBC or blood vessels, and nervous system disruption. 220 known bacterial toxins are damaged the 40% eukaryotic cell membrane. So this is the definition of toxins. Now how many types of toxins are there? There are two types of toxins which bacteria produces: exotoxins and endotoxins. First of all, we discuss about endotoxins. Endotoxins are basically lipopolysaccharide and known as bacterial poison. As you can say, poison of bacteria. Lipopolyglycans or lipopolysaccharides are mainly present gram-negative microbes. Lipopolysaccharide from gram-negative microbes does not necessarily require the destruction of the bacterial cell wall. The major component of gram-negative bacteria, it protects the membrane from certain kind of chemical attack and increases negative charge of the cell membrane and help stabilizes the overall membrane structure. Endotoxins are basically present in bacterial cell wall and extremely very toxic. First, this endotoxins discovered by Richard Friedrich Johnson's Perifer. Toxins kept within the bacterial cell wall and released only after destruction of the bacterial cell. That means endotoxins are produced or released only after the death of or destruction of bacterial cell. Bacterial lipopolysaccharide induces strong immune system. Now let us talk about the structure of these toxins. The structure of endotoxins, if we talk about structure, then endotoxins are made up of sugar molecule and lipid molecule. The sugar part is a repetitive glycan polymer contained within, and lipopolysaccharide is referred to as O antigen. Now, what is O antigen? O antigen consists smooth lipopolysaccharide and rough lipopolysaccharide. In rough lipopolysaccharide there is no antigen, and smooth lipopolysaccharide there is a O antigen. O antigen target for recognition by host antibodies and have more penetratable cell membrane to hydrophobic antibiotic. Another portion is core antigen. This core antigen is also made up of oligo sugar molecule and lipid molecule. Here, oligo sugar components that attach directly to the lipid A haptose and 3-deoxy-D-mannose octulosonic acid, also known as KDO, ketodeoxyoctulosonate, and lipid A portion are phosphorylated glucosamine disaccharides decorated with multiple fatty acids This act as anchor and lipid polysaccharide into the bacterial membrane responsible for much toxicity of gram-negative bacteria. So this is the structure of endotoxins. Lipid A might is very conserved component of the lipopolysaccharides. Lipopolysaccharides act as the prototypical immune system because it binds to the CD14, which is known as marker, TLR4, MD2 receptor complex, monocyte, dendritic cell, macrophages and B cells. These all cells and promotes the secretion of pro-inflammatory cytokines. Endotoxins are produced by the pathogenic bacteria known as lipopolysaccharide or lipoglycans. They are the part of cell wall, especially outer membrane, present in gram-negative bacteria. So here we discuss the structure of endotoxins and definition of endotoxins and how they produces their toxic agent. Now we discuss about the features of endotoxins. Endotoxins are encoded by bacterial genome. Endotoxins only present in gram-negative bacteria or produces only by the gram-negative bacteria. Endotoxins are fatal only in high doses. Endotoxins poorly antigenic, no vaccines work. Example, meningococcal and meningococcal toxins. Now if we look this mechanism of endotoxins, when endotoxins enters inside the host body, or this endotoxins is containing bacterial ingester or ingest by the host, the macrophages type of WBC produce or inform lysozyme cell. These lysozyme or macrophages produce IL-1 or TNF in macrophages produce cytokines. These cytokines involve in blood vessels, and when these cytokines IL-1 and TNF alpha present in blood vessels, then hypothalamus or pituitary gland produce prostaglandin release which cause fever. So this is the mechanism of endotoxins. Let me explain again. Endotoxins, when any kind of toxic agent, or we talk about bacterial toxins, especially endotoxins, ingest by the host inside the host body or ingest by host, when this endotoxins entered inside the body, the macrophages react and phagocytosis will be happen, and after phagocytosis IL-1 TNF alpha cytokines will be produced by the macrophages, and these cytokines entered inside the blood vessels, and these cytokines promote hypothalamus to produce prostaglandin hormone, and this prostaglandin hormone produce fever in host body or cause inflammation. Gram-negative bacteria makes an ideal molecule target for antimicrobial therapy. This is the whole mechanism of endotoxins. Endotoxins molecules Mimicry lipopolysaccharide molecules is thought to cause autoimmune based host response. Molecular mimicry is defined as the theoretical possibility that similarities between foreign and self peptide are sufficient to result in the cross activation of autoreactive immune cell by pathogen and derived molecule. In conclusion, lipopolysaccharide is most powerful weapon of bacterial cell, provide protection for bacterial cell but also confuse host immune system. So this is all about the endotoxins. Now come on the exotoxins. Exotoxins produces from gram-negative and gram-positive bacteria both. Exotoxins can be divided into following aspect based on the physical and structural activity. There are 5 type of toxins: neurotoxins, enterotoxins, cytotoxins, toxins that act from the cell surface, and membrane damaging toxins. These exotoxins act from the cell surface. Toxins bind to receptor on the cell surface and stimulate intracellular signaling. Example, superantigen. These superantigen are produced by bacteria like Staphylococcus or Streptococcus, cause toxic shock syndrome by binding MHC class II protein or antigen presenting cell. This activate T cell response with massive secretion of cytokines. And membrane damaging toxins disrupt integrity of plasma membrane. Example, alpha toxins from Clostridium species which cause gas gangrene. Alpha toxin has phosphorylase activity which enzymatically damage the membrane by removing the charges of polar hat group of phospholipid. Now, AB group toxins. These AB group toxins are intracellular toxins which passes A and B subtoxins. Example, cholera toxins. Different kind of disease caused by exotoxins. Example, Clostridium botulinum, toxic shock syndrome. This toxic shock syndrome, Staphylococcus aureus superantigen, this cause secretion of fluid and electrolytes from capillaries that decrease volume and lower the BP. This condition is called desequamated. In this desequamation, bacteria remove or outermost layer of skin. Another one is gas gangrene, which is caused by Clostridium perfringens. In this disease, membrane disrupting exotoxin myonecrosis is happen and blister will be caused in this disease. If we talk about mechanism of exotoxins, start when bacteria enter inside the host body. These exotoxins produce A and B cell and this A and B exotoxins attached to the membrane, surface membrane, and endocytosis, or which is known as receptor mediated endocytosis will be happen, and it target the ribosomes and affect the protein synthesis of host body. Now, how these toxins enter inside the body? There are some barrier present in human body or host. These barrier like skin and mucous membrane. The skin having a thin layer and 2.5 pH, which is acidic and very low, maintained by sebum. Dermis cell arranged very tightly because keratin protein, so that the intracellular junction is very less. Bacteria break through epidermis and have entry point through